Hello and welcome to the 10th presentation in the Treesearch and WWSC Science Center virtual conference series. This conference series was initiated to contribute to the scientific discussion in a time when conferences have been cancelled due to the COVID-19 outbreak. The full program is found at the Treesearch webpage. My name is Josephine Legård, and today I have the pleasure to present today's speaker, Anette Larsson, who will present cellulose derivatives controlling the drug release rate from oral formulations. Annette is a professor in pharmaceutical technology at the Chalmers, and she's one of the PIs in WWSC Science Center. And she was also the director for the Competence Center Sumo Biomaterials and is the director of the new center, Fiber. You in the audience can interact and ask questions or comment via YouTube chat functions. For that, you need to be logged in. Any questions will be answered after the presentation that will last approximately 30 minutes. And with that, I hand over to Annette.
Thank you. Thank you, Josephine, and thank you also for the invitation to give this presentation. And as Josephine said, there has been a lot of conferences cancelled. And one of the conferences that was cancelled was the ACS conference. And on that, my intention was to give this presentation on a section that was aiming to honor Ann-Christine Albertsson. And I asked Professor Albertsson if it was okay to tell you about the first time I had contact with her. And then we need to go back 24 years. And then I had just graduated as a PhD and had got the position at Astra Hässle at that time. And one of my responsibilities when I started there was to develop an oral vaccine based on polymer particles, solid oral polymer particles. And one biodegradable polymer is polyanhydride, and Ann-Christine is really an expert on that. So we were reading the literature and we came through a lot of questions. So we decided that we should phone her. And you know, I was young and I was so totally nervous, super nervous when I should phone Ann-Christine. And at that time we had this old type of phone, so we dial the number and we phone. And the thing comes here now that we was not just myself, it was Erik Söderlin as well. So we needed to talk with her both. And at that time at Astra, there was no speaker phones. At least we had not access to one. So we needed to sit like this together and shout in the phone to have her here. So that was before we did know the word social distance. But Ann-Christine, I think she realized that I was super nervous and she was very kind and contributed with her enormous competence and answered all questions, probably very stupid questions. But at the time, we were also working, and Ann-Christine had also been working a lot about controlled release and drug release. So this week will be handling about both cellulose derivatives and combine that with drug release. And the results I will present, most of the results are coming from my period in Sumo Biomaterials, which was a competence center around molecular transport, like drug release, in soft-based materials like cellulose derivatives. So, and I'm so fascinating of cellulose derivatives. The reason, there are two reasons. The first is that you can build in enormous much variety in the cellulose derivatives. By changing the substituents, how many substituents you have, or the molecule weight, you will have enormous different properties. So you can tune the properties by changing something. The thing is that when you do that, when you change something here, you know that you will change the properties, but it's very difficult to predict what the results will be. So today we are lacking knowledge when it comes to, for example, if you change the one substance to another substance, what will that give for influence on the thermal properties, the interactions with water and so on. So there will be a lot of more results needed. And I hope I can share my fascination over cellulose derivatives during this half hour, and also show a little bit about how different structures influence the properties when it comes to drug release formulations. And then in the title was also oral controlled drug release, and why do we work with controlled release formulation? Yeah, here we have is the blood plasma concentration and the functional time, and what we want is to have all the drug concentration in the blood plasma should be here in this therapeutic window. If you have a drug that has a certain lifetime in the body, then that is quite short, then you need to take a new formulation, new tablet or a new capsule or something like that, and then you will have this pulsing. And it also requires that the patient take a lot of different tablets during a day. So if you could give a formulation with controlled release, then you will achieve a more constant blood plasma, which will give you less side effects, but it will also reducing the number of dosing. So the frequency of dosing will go down. So this is really big advantages and helpful for the patients. And the most popular type of oral controlled release formulations is these two. We have the coating system, where you have the drug inside and then you coat it. So you hinder the drug to pass out as fast as possible. So the coating here controls the drug release rate. Or you have matrix tablets, and the one I would focus on is the hydrophilic ones, where you have something that dissolves, the carrier here dissolves in water. In this case, the drug is embedded here inside the tablet. And if you go to the literature, you will find that HPMC is the most common material that you have as a carrier. And so HPMC stands for hydroxypropyl methylcellulose. So there you have, so it is a cellulose derivative that is substituted with methyl groups or hydroxypropyl groups. And when you have this type of tablets, you will the water will go in and penetrate and start to dissolve the carrier material. And we will come back to that recently. So here is an example where we have had a rotating disc. The blue here is a disc that we have glued a tablet on, and then we have rotated it. And this has been done in an MRI equipment. And you can see here around that you have a gel starts to form. And if you take now this and shift it around, then you will get this picture here, which is a little bit clearer. So we have something that is called a core that contains very little water. And here, what we call a gel is not really a gel. It's a polymer solution with a very high concentration. And they have a concentration gradient here. And in between what we call the gel, pharmaceutical is called a gel, and the bulk surrounding it is called the erosion front. And if we then take a look here, the water goes in, and the reason why water goes in is that it will gain, since it's a hydrophilic polymer, the system will gain energy if it dilutes, conformation entropy in that case. And when the water goes in, the drug will dissolve in the gel layer and can diffuse out. This creates that you have a core with constant concentration of HPMC, and then it decreases. And here, the thickness of the gel we can call that H here. If you then go to the fixed law here, you can see that the water transport or the drug transport, it doesn't matter. If we simplify it a little bit, we can say that this is a diffusion process. Of course, it is based, if we go to details, that it is based on the gradient in chemical potential, where you have both influence of the osmotic drug release or transport phenomena or a diffusion process. Here I will just limit myself to describe it in terms of diffusion process. And then we have take the next here. So we take this now, we have the gel layer here, and our group was the first group that could demonstrate that we have a critical concentration here on the surface of the gel. And that is depending on the drug release that depends on this critical concentration, which we could show was proportional to the overlap concentration. And the overlap concentration is for entangled polymer solution, the concentration where polymers are entangled, but when you dilute them, water penetrates into the gel more, and they came more and more far from each other. And then they will reach a point where they are separate and have no contact with each other. And that is corresponding when they lose contact with each other is the overlap concentration. And that is depending on the size of the polymer chains, which then can be described with the radius of gyration. And we know that the radius of gyration is a function of the molecular weight and the persistence length. So if we have the increased the molecular weight, then the overlap concentration will decrease. And if we decrease the overlap concentration, then more water needs to be transported into the gel. So if we go back here, more water needs to go in here and dilute the gel more and more, which means that you have a longer, larger gel layer and a longer diffusion length there, which then slows down the drug release rate or the water transport rate. The persistence length is also controlled by the polymer properties. In this case, the interactions with the water will rule it, and then it comes to the degree of substitution. So this can explain why the molecular weight and the degree of substitution, which has been shown for a long time, is controlling the drug release rate from this type of formulations. But here comes an example from our group where we have the polymer released as a function of time. These are 7 different batches of polymers. And in pharmacy, it's very critical that all stuff you use is pharmaceutical approved. So when to get it pharmaceutical approved, you set up a criteria, what parameters that you should test, and they are tested for all batches. So when they are pharmaceutical approved, they should be the same. That is the behind thinking about it. So this is just nothing else. It's just a polymer, and you can see that this polymer is eroded away after 20 hours, and here it's not eroded away even after 100 hours. And we investigated these 7 different batches, and we could see that this was the same molecular weight and the same degree of substitution, which then makes a little bit question, why does this happen? So we continued and start to look, think about it. And of course, we did know that the substitution pattern for this type of heterogeneous materials are very critical. And there are different types of substitution patterns you can talk about. It can be within a glucose unit. So here you have the 3 possibilities where the R group can be changed to an H, and then it's not unsubstituted, or if it can be a methyl group as it is one type in the HPMC. And they can be on this number 2, 3, or 6 position. And we will always have some kind of heterogeneity when it comes to where in the glucose unit it is substituted. So here I will give an example from Professor Thomas Heinz's group in Germany, where Katrin Fletcher and he showed for CMC that if you have a low degree of substitution, 35 here, then you can see that the number of substituents on the 2, 3, and 6 position is about the same. But when you increase the number of substituents, then you can see that you will have about the same number of substituents on the 2 and 6 positions, but much less on the 3 positions. So this position is less substituted. That is, of course, very important information, and that will give effects on the properties of the polymers. Here's also an example where you, for example, if you take 1.3 total degree of substitution, as we have here, they show that there are a combination of this line here corresponds to when you have no substituents, so about 25% or something like that is unsubstituted. At the same time, you have some that are, the majority has 1 substituent, but there have also some that has 2 substituents and some that has even 3 substituents on the glucose units. So this shows that the substitution pattern within a glucose unit is very critical. What we was focusing on was the substitution along the backbone. So here's an example of that, where the dark one symbolized where you have substituents, and the white one where you have unsubstituted glucose units. And if you take and treat these with enzymes, then the enzyme can cleave if they have unsubstituted units aside the cleaving point. So it can depend, different enzymes have different degradation patterns. So there can be 2 or 3 or 5 unsubstituted units required to just chop off 1 glucose unit. So what we did was that we could correlate the degree of substitution, the heterogeneity in the substitution, and that was measured by how many glucose units without any substance we could find when we treat them with enzymes, and we could compare that to how many glucose units without any substance we had when we had acid cleavage, so we cleave them by acids instead. And on this axis, we have the T50 value, and that means when 50% is released. So here, for example, the T50 value is around 45, corresponding to these points. And you can see that the slow ones here has the highest degree of heterogeneity in the substitution pattern, meaning that the most glucose units was cleaved away. And if you want, you can see for more information this publication here. But then comes the question, okay, so it depends, a slower release is gained when you have more heterogeneous materials, but why? Why is that this happen? Is it due that you have some interactions between the substituted pattern, or is it due to that you have interactions between unsubstituted ones? And to check that, we did SEC measurements. So this is size exclusion chromatography, where we have the IR detector and we have a multi-angle laser light scattering detector, giving an indication of the size of the things that enters to the detector. And at low temperature, you can see very quite monodispersed stuff. And in SEC, the longest one comes first. So if it higher the temperature in the SEC column, on the SEC column, you can see that we got the new peak here, which is an indication of that we have got aggregation, that we have somehow built up materials at higher temperature. And if it should have been interactions between unsubstituted areas, then we shouldn't have expected this type of temperature dependence. But it is current with the temperature dependence that goes with that we have interactions between the heterogeneous parts. So at higher temperature, more heterogeneous, the heterogeneous batches was interacting more. That was also showed by, we were also making phase diagrams in another publication by putting a centrifuge in the 80 degrees oven. And I should say that this is an experiment that works quite well until the centrifuge breaks. But still, it costs a centrifuge. But it was good from that point of view that we could prove that this is really the case, that we have interactions on these batches, more strong interaction between these substituted regions. And this also influences, of course, the swelling. So here is an example where we have, this is the release curve. So how much that polymer that is released. So here we can say 40% is released, here's 100% released. And the erosion front is the front where we have the bulk gel interface. And you can see in this case where we have slow release, we had got a large degree of swelling, which indicates that a lot of water was needed to penetrate into the gel before we reached the point where we could have this reach the critical concentration. So that strengthened our theory. Now I should, so that was an indication of that the H, the thickness of the diffusion pathway, or what we can call it, that is very, have a very clear impact on the drug release rates. For other systems, like the coating system, where we coat it with polymers, then this has also a very strong release. So what we did here was that we took to the other cellulose derivatives. One was ethyl cellulose, which is insoluble in water, and this hydroxypropyl cellulose is soluble in water. We dissolved both of them in this case in ethanol and sprayed that on pellets. So we coated here, and during the spraying, we found out that we had a phase separation, and in this case, the phase separation was due to a Spinout decomposition. So when we have phase separation, we start in one phase, and we go when the ethanol, it was dissolved in ethanol in this case. When you have the coating process, then you dry the system. So ethanol is evaporated here, and then you get a higher concentration, and the polymers precipitate and form the coating. So what you start here is that you start in one phase region, and when you evaporate away the ethanol, the concentration of the polymer increases, and you reaches a two-phase region. And in this two-phase region, you will have one phase with this polymer concentration and one phase with that polymer concentration. And this is, of course, due to that you get the phase separation due to that you have a low contribution for polymers to the entropy contribution. But there are different mechanisms by the phase separation can occur. One is Spinout decomposition. So you start in one phase region and goes to this two-phase region, and then you have this thing here developing. You can see how it grows, and at some point it locks. And this picture is from Sofia Wassén in Sumo, and she was also working at RISE. But you can also have nucleation and growth, where you start in the one phase region, and you get to this where you have nucleus that start grow and grow and grow. And then you will get another type of structures, morphology in the end. And we could show that depending on, this is just to say that the entropy contribution for polymers is low. So we could show that depending on the molecular weight, the phase diagram was changed. So we could both get spinol decomposition on nucleation and growth, which gave a lot of impact on the controlled release. Because if you have individual domains where you have spinol and growth, then that is not connected to each other, then you will have very slow release. And the reason for this is that the drug release is controlled by that you have this coating layer, which has one phase of the EC phase and the HPC phase. And when we place it in water, the HPLC phase will start to dissolve, and then you create a pore, and that pore can be used as a channel for the drug to be released through. Another thing when you change the molecular weight is also that you lock the system at different positions. So we tested this with using two different ways of make the coatings. Here we made free films. So we had a rotating drum, and here was a traditional fluidized bed where we coated the pellet. And here is the molecular weight that changes. And here is the cross sections. And here you can see at low molecular weight, we have this. What is remaining here is the EC. So these films has been exposed to water, so the HPC has leaked out. So the holes here is where the HPC was before. So you can see that the structure and morphology is different here compared to the other ones. The pores are bigger here, and they became smaller and smaller. And that is then due to that the coating process was the system was locking, the phase separation, the spinol decomposition was locking at different positions, which gave them different morphologies. And then we can explain that by the two-phase things, that the phase diagram, the shape of the phase diagram changes depending on the molecular weight, but also that higher molecular weight will give higher viscosity, which locks the system at when less ethanol has evaporated, meaning that less phase separation, the occurrence of the phase separation has been stopped earlier. And we tested the permeability in these diffusion cells. So we took the films, placed it here in between two cells, and have tritium labeled water on that side, and looked onto how much tritium labeled water that received to the acceptor chamber, and we could take the slope, and the slope is then proportional to the mass transport and the permeability, and that is approximately equal to the efficient diffusion coefficient. And this diffusion process then will depend on things like the porosity. And one way to characterize the porosity is to determine how much of the things that give rise to the pores is you find has been released. And the HPC amount of HPC release is here determined. And you can see for the three lowest molecular weights, you can get out about 95% of the HPC of the total amount you have had for the beginning, but here you only get out 80%. This is due to that you have not so connected pores here, and it is also more difficult for the polymers to diffuse in more narrow polymer chains, so that takes more time, and therefore they can be locked into in the coating. So we can say that the permeability has a very strong dependence on the porosity, but here it comes another thing. Here you have the same porosity, but you can still see that the permeability decreases. And what we did was that we compared the permeability for water with the permeability for mannitol, vertically labeled mannitol. And then we could see that it was not just the porosity that was an important factor, also the torosity is an important factor. And porosity means the way, the diffusion pathway the molecules needs to take when it goes through the coating. Okay, so this was examples on the for freestanding films. We also tested the same if it was valid for the pellet system, where we have then the drag release, how much that was leaking out from them. And you can see here that the release takes longer time when we increase the molecular weight. So, and that is also due to the structure of the coating. So permeability is a super interesting thing. We are also now working with oxygen or gas permeability. So we have started to build our own permeability measurements instrument where we have the film and we flush through the gas on one side and measure how much oxygen in this case that has reached the other side through the film and determine that with the MS detector. And the long-term goal here is that if you have different cellulose derivatives here, can we predict the transport of oxygen, but also in presence of water, what happens then? And also can we get a better understanding on how different substances influences the thermoplasticity with or without presence of water vapor, or at different relative humidities. So this is an example of where we have changed the structures of the polymers, and then it influences the properties in the solution and solid state. But you can also change the formulations and the structure of the formulations, and do that can create, for example, different porosity or morphologies. You can change the size, or you can change the area volume ratio, which then directly influence the properties of the formulation. This structure property relationship here is very important, and also to many extent not fully understood yet. So here is an example on where we have ethyl cellulose, where we have mixed into a drug called philodecene, and here is filaments from work in our group. And you can see that this TG for these filaments decreases, and this is the Gordon-Taylor model. So it's not behaving ideal. And today, very little is known on why this philodecene interacts as it does with the EC. And we could also take these filaments and print them and make tablets and check the drug release rate that is at least possible to do. So the come up question here, what is the underlying mechanism for the thermal behavior of the EC in presence of drugs? Can you take another drug and then see if that behaves equally? We know that that will not be the case, but can we look at the structure of the drug and predict how the thermal behavior will be with different cellulose derivatives? So that is, and this is a little bit the drug in different, and the thermoplasticity and how that influences the cellulose derivatives is something that became more and more important, since this can be a way to make individual personalized tablets in the future, which is a future demand. And also that it can enable continuous manufacturing of the cellulose using hot melt extrusion. But the questions here around the interactions and the mechanism behind how that influences the cellulose derivatives, that is still unknown. And what we hope is that the competence center fiber can help us to understand better about how we can get materials, thermoplastic cellulose-based materials. So we hope that the center can contribute with knowledge that will, in the long term, make it possible to get a transformation from fossil-based plastic to lignocellulose-based thermoplastics. And we are aiming to use 3 different routes here, and they should work very closely together. A tree without roots will not function, and a tree, if you take away one of the roots, will behave much less good, feel much less good. So we hope that or we're aiming to that the molecular modification, advanced characterization, and predictive processes should work close together. And then they can give the flowers we have apple trees here in the garden that starts to flowers. So we hope that these trees can flowers, and we have in fiber companies that are through the whole value chain here. And we hope that the center can contribute with knowledge to the in between here, so both knowledge from the companies to the academia and vice versa. So that is a long-term goal here. We have just started, so I couldn't show any results from that. The first PhD project started about in April, so it's no results yet. But here is a little bit of the companies. So we hope that we can deliver the knowledge we need to take this step change, which will take a lot of time, of course. But it is, as you see, a lot of partners, and we are very happy for working with all of these partners, and both PIs and partners. So yeah, I have a lot of fun, and I hope we can contribute to do good research. So my conclusions is that cellulose derivatives is one part that opens up for a large variety of functionalities, and you can even tune the properties of them. The problem is that you don't know, you can't predict yet how to tune things. But with increased knowledge, I hope that we can get better potential and more knowledge about how to use the cellulose in the future for a lot of different applications. It can be drug release, but it can also be for packaging and yeah, wherever they are used. So by that, I want to thank the slides I've shown is based on these contributing PhD and postdocs. And I want to thank the other co-authors I have been published with, and Thermo and Fiber, and also the financial support, and especially the companies here that have directly give support to this, what I here presented. There are a lot of other companies and partners and people I would like to thank, and I will also thank you. With that, I'm ready, Josephine.
Thank you very much, Anette. And now we open up for you in the audience to ask questions via the live chat function. I know that there is a small delay, so I will just say that very interesting work, Anette, and it's so interesting to see that your research is really combining pharmaceuticals and then more about this forest, more was to say traditional forest industry research, polymer. Is there something that we can learn from the pharmaceutical research?
Mm-hmm. Yes, absolutely. I mean, for me, the drug delivery is an application, and I'm fascinating of the materials there. How can we tune them? How can we control them in better ways? How can we characterize them? And in the pharmaceutics, as I said, there are a lot of rules when it comes to approving new pharmaceutics, pharmaceutical excipients. So that I think the techniques that is used for in the pharmacy for cellulose derivatives can be applied in the cellulose. But I also think the other way around, the knowledge around of on cellulose structures and yeah, how you dissolve cellulose and stuff like that can also influence and in the future also make better tablets. So I think it is giving and taking from both sides.
Mm. And there's still opportunity to write in questions. I take the opportunity to say something about the future presentations in this series. So now on Tuesday we have Peter Olsen from KTH, who will present the synthetic strategies for decorating biopolymers with rings. And that will be on Tuesday, 10:15, and broadcasted through the YouTube channel. And after that, let's see, we are approaching the end of this conference series. We will have a presentation on the 26th of May with Professor Monica Ek at KTH, and the 28th May with Marina Hassani from Chalmers. And you can find all the previous presentations on YouTube. And if you want to rewatch Annette's presentation, of course. Now we have a question for you, Annette.
Oh,
it's Jakob Redingepon. Says, thank you, Annette, for your interesting presentation. You have shown examples on prolonged release. What about increasing dissolution? See carbon derivatives. See, I lost my window there. Cellulose derivatives as disintegrants, is that concluded?
That they are cellulose derivatives are used as disintegrants, for example, Cosling CMC. But I should say that there have been lots of research when it comes to solid solutions and solid dispersions, which I didn't mention that, but that is another area where you take the drug and you want to have it in amorphous state to increase the dissolution rate, and then you need to stabilize the amorphous state, both stabilize it in the solid state, but also when the material is dissolved, so it doesn't recrystallize back. So, and then cellulose derivatives has been used for that. And for example, HPMC AS, we have used it and also looked into how that can inhibit the recrystallization. That was actually done also in my major tablets. But the thing is that if you have hydrophobic parts of the cellulose, then you can inhibit that can interact with the more hydrophobic drugs in this case, and then you can get inhibit. So that is a way to create an artificial higher solubility, so apparent solubility by keeping them in a supersaturated state.
So thank you. We have no more question.
That's good. And if you have questions, just send an email to me or Josephine, and we can try to answer them as good as we can.
So thank you very much, Annette, and thank you to you who have been listening. So with that, I close this presentation.
Okay.
Session.